Hi, to conclude our videos about membrane proteins, we'll talk about channels, and we'll talk about it primarily in the context of pumping, allowing ions to flow across a membrane. We'll talk specifically about how channels can be gated, that means opened and closed, in response to some regulatory event. We will study in molecular detail the potassium channel, and we'll show how charged ions can first be stabilized in the hydrophobic membranes, and the amazing yet straightforwardly simple selectivity filter that can prevent smaller ions like sodium to get through. We'll get introduced to the technique of patch clamping, which is how scientists study the kinetics and dynamics of ion channels, and then we'll talk a little bit about nerve conductance, which relies on a series of ion channels to propagate an action potential. So ion channels are both selective and gated, meaning selective, they limit what can pass through it, and gated means that they can open and close in response to a specific stimulus. It can be a binding event, it can be a voltage change. In general, channels do not open halfway. They have two states, they're either open or closed. And because they're sort of a passive highway for ions to go through, the rate of transport is much higher than transporter proteins, which we've seen in previous videos. This slide shows various ways to gate an ion channel. The one which we'll talk about most is voltage gating, and that is when there is a change in electric field in the membrane near a protein that will cause conformational changes in the protein to occur. And in actuality, very small protein changes can make large differences in pore size. So again, there's nothing mysterious here. It's straightforward electrostatics and thermodynamics. Ligands can gate channels. They can bind to and cause conformational changes opening channels. That can occur extracellularly or intracellularly. And then there are mechanically gated channels that open in response to a pressure, such as ion channels on the hair cells in your ear, and also the ion channels which signal pain or touch in your fingers are examples of mechanically gated channels. It was a major event when the potassium channel was crystallized and its structure solved three-dimensionally. This is a cartoon of what it looks like, but it's going to highlight a couple of important points. The first thing was, if you look at the electrostatic potential and where ions would not like to be in a membrane, the center of the lipid bilayer is the maximum place of where ions should be unhappy. So the question was, even if there's a pore, how are ions stabilized in this area of a membrane? And the second question was, whereas it is conceivable why potassium ions, which are larger than sodium ions, why potassium ions would not be able to flow through a sodium ion channel. But as far as I know, no one ahead of time predicted the mechanism whereby a larger pore, which would allow the passage of the larger potassium ion, could still prevent the passage of a smaller sodium event. And now that we see the answer, it's almost impossible to imagine that at one point we didn't suspect this. So let's get to the first point. How does the potassium channel stabilize potassium charges in the center of the membrane? So first of all, potassium ions are free to flow into the pore on this side, and they are stabilized here in the center by the electric fields produced by these alpha helixes. Remember when we talked about enzymes, we talked about the dipole moments of all the individual peptide bonds and how those vectorially sum to produce a larger dipole moment associated with an alpha helix. And these focus negative fields, negative electrostatic field in the center of this vestibule area, which can help stabilize potassium charges. For the selectivity filter, we'll go to the next slide. And now we'll show two views here. We're going to show two cross-sectional areas, and unfortunately they're crossed, but we'll look at the state of the potassium and sodium ions in the vestibule. Those are surrounded by water. These are hydrated ions, so that will be shown up here. And then we'll look at the ions in the selectivity filter down here. So first let's look about hydrated ions in the vestibule or out here in solution. A positively charged ion in water solution is going to be coordinated by at least four water molecules. Remember, there are partial negative charges on the oxygen atoms, and these help solvate or stabilize a potassium ion in solution. The same is true for sodium. The size difference matters almost nothing. The smaller sodium ion might have the water molecules separated by a slightly smaller distance. But now let's look in the selectivity filter. Again, if the potassium is stabilized by four water molecules here in the vestibule, for it to flow in this direction, it has to have a approximately equal degree of stabilization. Otherwise, it would be energetically unfavorable to move up in the selectivity filter. So how does the protein do that? Well, it's got a fixed geometry of carbonyl groups, which can coordinate with the potassium ion. Again, partial negative charges here, lone pairs of electrons and oxygen atoms, and help stabilize the potassium ion in the selectivity filter, just as the water molecules did in the vestibule. But the sodium ion is smaller, and here the position of the carbonyl groups are fixed. In solution, the water molecules can approach the sodium ion as closely as necessary, but with the fixed positions, you're not going to be able to simultaneously coordinate all four oxygen atoms, so this is energetically very unfavorable. Here in the vestibule, the sodium ion is stabilized by four water molecules. Here it is stabilized by only two carbonyl groups. Thus, sodium ions will not want to, they might hop on once to the first one, briefly find out that it's energetically unfavorable, and then they're going to have to decide whether to go that way or this way, and thermodynamically, most of the time, they're going to return back to the vestibule. I'm using language that anthropomorphizes a little bit, but there's no decisions making, it's just thermodynamics. The most frequently utilized method for studying ion channels, which is amazing in its own right, is a method called patch clamping. There are several different ways of doing it, but the fundamental principle is that when channels are open, you can have a current flowing, and that can complete a circuit, and with sensitive electronics, you can measure the current. And there are different ways of doing this. You can do whole cell recording by putting an electrode up to a cell. You can apply a pipette, a very fine pipette, again micron in diameter, to a cell membrane, and with suction very gently pull off a portion of the membrane. Again, this would be a circular patch shown in cross section here. And in this example, there are the schematic shows two ion channels present, but you're also able to study single isolated channels. So this is a macroscopic method which is capable of measuring current through a single molecule, which is really amazing. And here is a slide that purports to be real data, which I assume it is. The opening and closing can be stochastic, but again, emphasizing that a channel is either open or closed. If it's a single channel, you won't see stages like this, and it's always a series of step functions. Unless you have many channels, then you'll never see like a smooth increase in the amount of current that's flowing. And if you try to define a characteristic time that these channels are open, it looks to be approximately about a millisecond. Now, axons rely on ion channels for the action potential propagation. Here we have the soma or cell body of a neuron, and here are the dendrites or the nerve terminals. This distance can be up to 3 feet. The largest nerve in your body is the one exiting your spinal cord, in your sacrum, and extending down into your big toe. And so based on electrical inputs to the cell body, a summation signal will occur, and if it decides to start, if it sums to start an action potential, then you'll get an action potential proceeding all the way through, down through this nerve, and there'll be some biochemical event here at this nerve terminal. When you think about how quickly you can respond from stubbing your toe to experiencing pain, you know that that can happen in, I don't know, milliseconds. You can appreciate that the velocity of an action potential, which is our molecular changes, can occur quite quickly. So an action potential is properly referred to as a traveling wave of electrical excitation. Now, you will study this in greater detail in your systems physiology course, but we'll talk a little bit about the molecular events here. We'll focus primarily on the sodium channel, and in systems phys, you'll learn about all the other channels that play roles here too. At a time 0, you have a channel that's open that's producing an electric field disturbance around or change around the ion channel, and that will trigger its next door neighbor, which has got to be close enough so it experiences the altered electric field around the open ion channel. That will trigger this to open. When this channel opens, now ions are free to rush through here. After a short amount of time, you'll have produced a large electric field change at this location, and that will in turn trigger the opening of a third ion channel, and so on down the axon. Now, action potentials only propagate in one direction because there is a refractory period. Once a channel is open, it closes and it's not able to open again for a while. By the time it's ready to open again, this traveling action potential has moved several channels away, and now when it's capable of opening again, it's no longer near the electric field produced by the open channel. And this schematizes what we just said in a little greater detail. It's even trying to indicate the relative strengths of the electric fields. Now, the point I want you to consider is to think about 2 successive pinpricks to a particular body location. If 2 pinpricks come very close together, they will feel like a single pinprick, and then if they come 10 seconds apart, they will clearly feel as two pinpricks. So think about the amount of time that can elapse between successive pinpricks, but the shortest amount of time where you can still feel 2 pinpricks. It's definitely less than a second, and I don't know what the answer is, but the point I want to make is think about how much sodium and potassium pumping needs to go on to get 2 action potentials. So you have 1 action potential, you've got all this sodium, all these sodium ions flooding in, and now you have to pump that all back out in order for the second pinprick to produce another action potential. And you can start to appreciate why ion pumping with transporter proteins is such a huge energy expenditure, especially when it comes to with respect to nerve cells. We'll later think again about the speed at which these molecular events need to occur when we talk about muscle contraction and the opening of calcium channels, and then the pumping of calcium out, the opening of calcium channels and pumping it out again, and how fast they have to go for you to flex your elbow and move your hand from a fully extended position to a contracted position in about a second. So here we see a synapse, and there's lots of interesting biology going on at a synapse. And this postsynaptic cell could be another neuron sometimes, it could be a muscle, it could be any sort of tissue that does something in response to nerve stimulation. Here's what's going to happen. You have the action potential coming down the axon to the dendrites, and it's going to produce a voltage change which will eventually cause a voltage-gated calcium channel to open up. This allows calcium ions to flow into the end of the nerve, which is called a bouton, and that will be the trigger to cause this synaptic vesicle, which will sit here as long as it takes until an action potential comes along, but this calcium is triggering it to fuse with the membrane. This could be done by calcium binding to proteins that maintain a particular shape of the vesicle. And now the neurotransmitter molecules that were contained inside the vesicle are released into the synaptic cleft. They will diffuse across the cleft and can bind to the channels on the receptor cells or postsynaptic cell. This will open the ion channel. Ions can flow in or out, and this has the potential of causing an event, which could be another action potential on another neuron. So you have voltage-gated sodium channels and potassium channels causing an action potential, a voltage-gated calcium channel opening to trigger the fusion of a vesicle with a membrane. You have the diffusion of neurotransmitter ligands into the synaptic cleft, binding of another channel, gating it open, allowing ions to flow through, and another action potential to potentially occur. And then on this slide, we have a real image of where in red is stained synaptic clefts, regions where the terminal branches of other neurons are making contact to the body of another nerve cell, which is schematized here in this picture. You have the axon, so action potentials for this neuron will propagate down through this way, and we have dendrites here that are collecting information from other cells and a lot of other neurons which are interacting with this neuron. So in this example, there are hundreds to thousands of inputs to this neuron to allow it to make a binary decision to either send or not send an action potential down its axon. And maybe binary is a little simplification because they can send action potentials at different frequencies, but still it's sort of awesome the way the neurons are able to integrate so many inputs and to then base its behavior on an evaluation of the various signals that it's receiving. All right, well, this concludes our section on membranes and membrane proteins and membrane protein functions. Thanks for listening.